So a lot of great ideas and thoughts coming through on this. I also want to thank Aaron because he gave a great introduction to the microRNA, so I don't actually need to spend any time at all introducing the concept. But what I want to do is perhaps just a slightly bit difference in the next, I guess, about 40 minutes is what I was allotted here to talk. And it's really to return back to my first love, which was really to understand neurofilament metabolism in the motor neuron degenerations. And more specifically, over time, we've become interested in what regulates the low molecular weight neurofilament RNA stability. And I'm going to focus just on two particular pieces at the moment because I don't think you can look at RNA binding proteins and microRNAs as separate entities. One has to think about them in a sort of a consensus type fashion. We haven't talked an awful lot about one of the primary pathologies of motor neuron degeneration, and this is really what got us started many, many years ago in this, and that is the traditional pathology of ALS is a motor system degenerative disorder. Now, we've clearly moved that over time, but when we look at the motor neuron pathology that exists, the classical pathology is indeed a number of intermediate filament inclusions. So here we're looking at neurofilamentous inclusions, marked by SMI 31, and they take quite a range. Everything from these serpent-like, which they were called at one time, we now just call them neuronal cytoplasmic inclusions, to some that are a little bit more crescent-like or vacuolar in their nature, to these large aggregates which can sit within the cell or actually sit out in the neuroaxonal process, at which point they're called neuroaxonal spheroids if they're over 20 microns in diameter with that. But we also see that peripherin can do exactly the same thing. And alpha-internexin, although the literature holds that alpha-internexin is never deposited in these neurons, here's a pretty good example of a massive alpha-internexin aggregate, probably replacing nucleus, but we don't know that for sure. And in the original work, we all considered this to be important because we could mark it either with ubiquitin or P62, which suggested that these proteins were into a degradative pathway. And so this was the classical pathology as we all taught it up until almost a decade ago. And now the problem is we also have this contemporary pathology that we need to reconcile. So this is also human spinal motor neurons. And what we're demonstrating here is a variety of different RNA binding proteins, all of which can form aggregates within spinal motor neurons in ALS. So we've now got a contemporary pathology, which we've obviously been talking about for the last day or so, and the more traditional pathology. And so how do you reconcile them? And to give a brief history as to our interest into that and why we focused on one particular area, one actually has to go back to the early 1990s. And a Canadian researcher, Catherine Bergeron, was the first to demonstrate that in aggregate-bearing motor neurons in ALS, when she measured total poly-A-RNA and compared it to NEFL message RNA, she showed only in the aggregate-bearing neurons that there was a reduction in the NEFL message RNA species. A few years after that, we asked the question, well, what would happen to all the other ones? Because we know that, for instance, vimentin can come in, or alpha-internexin, and actually form the initial homopolymer on which the ultimate heteropolymer of neurofilament is formed. So where are they in this equation? And what we found in spinal motor neurons was that if we looked at NFL, M, or H, and this is control versus what we were seeing in the ALS population, we saw a significant reduction in the NEFL message RNA compared to H or M. And then we also saw that both peripherin and alpha-internexin were reduced, so the compensatory ones weren't there either. So it's just changing at that point what we thought was the stoichiometry of expression of the RNA species. And why is that important? And it goes back to just understanding that these are a heteropolymer that are required for the formation of the neurofilament triplet. And that neurofilament triplet is then critical for, initially in our thoughts, for cytoskeletal structure, but as we're beginning to learn more and more about them, also for cytoskeletal dynamics and for anchoring, particularly the presynaptic membrane with those. But the key piece in all of this is that we have 3 of these intermediate filament proteins, so low, medium, and high molecular weight. We're not all that creative in the protein world. And they have this large C-terminus domain, and as you get to H, much larger with a phosphorylation domain. And for the assembly, the first thing that you require is that you must homopolymerize the NEFL. right? So that needs to form a dimer. And then onto that is layered either H or M. And the side arms swing out. So there's some beautiful work from French group a few years ago showed that for NFH, depending on the degree of charge there, the C-terminus domain would actually swing out from the side. And there's about a 12-degree radius that it will go through. And as it goes through that, you electrostatically change the interactions between the filaments, and it's thought that that helps control the axonal diameter. So phosphorylation is important, formation of these is important, and getting to this in the first place is really important. And depending on what axon that we're looking at, the packing density is going to be different between those. And so for motor neurons, the argument has always been that the density of these individual filaments, here we see it on cross, is so high that it places them at risk. So it is one of the explanations historically that's held. So we were really curious as to why was it that we and Pam Shaw and Catherine Bergeron before that had seen this difference in expression and what we had seen as an absolute reduction in motor neurons by in situ hybridization. And so we did a cheap and dirty experiment which got us into an awful lot of trouble for many years with reviewers to try and explain. And all we did was we expressed the human neurofilament messenger RNA in a test tube, and then we added to it homogenates of human spinal cord post-mortem with that from ALS or control patients, and then simply measured the stability of the RNA, so how rapidly was it degraded over time. And in contrast to what we saw in single cells, in fact, when we exposed the RNA to ALS spinal cord, it was actually more stable than when it was exposed to control. The exact opposite, right? We'll come back to explaining that later. For that, we could boil both of them, and actually we increased our stability, and we also brought the control up with that. We could use a proteinase K, and we could take the ALS sample, digest it, and bring the RNA stability up to that of control with that, all suggesting that this was likely protein compounds that were associated with the homogenates that were actually bringing about the stability determinant for that. And then in a simple experiment, we simply changed the amount of either 100% control or 100% ALS homogenate in there, and we could shift the stability of the RNA species back and forth with that. So that was our first clue back in the early 2000s that we were in fact dealing with some protein interaction which would lead to a stabilization for that. And now I'm going to skip a decade of an awful lot of work of describing, and we're going to move to a relatively straightforward or an interesting example of how this stability could actually be determined. But there are, at least, and I was commenting to somebody at the break, that when I'm teaching students about this, I tell them, well, there's only really 3 ways to change stability of an RNA species. Once it's already in place. Well, there's more than that, but to clump them. One of them is to pathologically express a normal RNA stability determinant. And we're going to talk about one that I don't think you've heard much about, and that would be RGNEF. So one of the nice things is that, and again, some of us were talking about this, for the students in the crowd, if you're going to pick a research topic, pick one nobody else is doing and then stay with it, because nobody else will do it either. And so you can create a niche. And so these are all of the RNA binding proteins for neurofilaments, the H, M, and L, that are known to date, the last one being described in 2010. And that was our RGNEF component of that. And since then, we haven't had any additional ones appear in the field. So it makes it relatively easy, but I'm also going to show you a slide I use to depress all of my students at the speed of light, and that's this one. So this is NEFL mRNA, right, with the entirety of the 3' coding region here, right? Half of it is regulatory, right, in terms of that. And in our lab, we work right here. And that's going to be real important when we come back to it later. But we have to remember, when we talk about binding sites on an RNA species, we're talking about one single site amongst an entire complex structure that's going to change depending on the binding. And that's going to have some role in that. So what is RGNEF? Well, RGNEF is the human homologue of P190-ROGEF. P190-ROGEF was described by Bill Schlepfer and his group back in the late 1990s. And it's a critical determinant of NfL, murine NfL stability. Mutated, it leads to a motor neuron degeneration. And for years, we worked with Bill to say, why don't we look and see if there's a human? And Bill said, well, why bother? So eventually, we did it on our own. And we found that there is, in fact, a human homologue of P190-GEF, which is called RGNEF. Rho-guanine nucleotide exchange factor. It has a RNA binding domain, and I'll come back in a moment and show you some additional domains to it, which shares about 76% homology to the mouse. And then it also has a GEF binding domain, or GEF activating domain here, which is almost 92%. And this is going to be a very important feature as we move forward with this. Because when we look at the protein, it's huge. It's about 145 kilodaltons when all is said and done. And it has some really intriguing domains that we are going to repeatedly return to because they are important for us. So if we start here at the N-terminus domain, we have a leucine-rich domain. We've got a big area of Lord knows what. And then we've got a zinc-binding domain. This plextrin and double homology domain is really important for GEF activation. And I'll spend a second on that, and then we'll leave it. And then there's another binding domain here for FACT. But what's really interesting is it has an RNA binding domain right in the middle of a coil-coil domain. With it. And then it also has a PDZ binding motif out here. And all of this is also overlaid by a microtubule binding domain. So as we've heard in the last 48 hours or so, many of these proteins are pluripotential in terms of their interactions, and this is no exception to that. What is absolutely unique about this as a RhoA activating protein is it the only one that we know of this far that does both RhoA activation and RNA binding capacity for it. There are no others that we can find in the literature, and that's a fairly exhaustive search with that. So in a rough schematic of how we think that this at least is being assembled, and this is just purely a schematic for grant purposes, is that we do have two domains. We have one which is really Rho activation, and then we have a second which is RNA binding and regulatory. And I'm spend the bulk of my time on this, with the exception of one slide, which is to prove, in fact, that it does actually activate a guanine exchange factor pathway and RhoA activation. And to do that, the assay is simply that an inactive RhoA contains GDP. You have to exchange a phosphate and put a GTP in place for that. And you can actually measure that by looking for phospho-RhoA and comparing it to the non-phosphorylated. And indeed, when we express guanine exchange factor in vectors for that, we actually see a marked upregulation of the phosphorylated form. So that's all I'm going to say about that GEF activation domain. We're doing a lot of work on it, and it's turning out to be a challenge in terms of where it sites and functions, but it is a GEF activating. The rest of it, however, the important piece, is to really function on its interactions with NFL message RNA. And in this study, what we've demonstrated is that at the time we didn't have antibodies. You'll see those in a moment. So we just created a MIC-tag RGNEF, or we deleted out the RNA binding domain. And what we showed was that in the presence of the full, we could actually immunoprecipitate NFL. So this is an RNA IP. You immunoprecipitate with a protein first, then you do a PCR off of that, see what RNA has come with that. But when we delete out the RNA binding domain, we get nothing in there. So that suggested that, in fact, there was an interaction. It doesn't, in fact, tell us that it's functional. So an assay we're going to return to several times is a luciferase assay, and it's been referred to a couple of times in talks. I'm not sure that everybody knows how these are done. So this is done for microRNAs, but it's an example of that, where what you do is you take, in this case, our 3' UTR, and we attach it to a reporter, and it's a luciferase reporter for that. And then you bind your target to it, and then you measure the amount of fluorescence that comes off of that. So the amount of fluorescence, whether it increases or decreases, tells you whether you're stabilizing or destabilizing your construct. You need to always marry that to a PCR product to ensure that you, in fact, are maintaining this or destabilizing it, depending on what's there. So when we did that, we ran into a second problem. And as you're going to discover as I go through this talk, that I'm giving you a lot of problems and not necessarily a lot of answers for them. So our next problem was that when we started to look at the NFL message RNA in the human spinal cord, we started to come up with this, at least three isoforms. All right, so we've got one which is, and this is just the 3' UTR, right? So about 430 some odd bases sitting in there, right? And then we have an intermediate one, which runs a little bit higher, around 1,300. And then we have this much larger one that runs about 1,800. So all the black is where we have found these in human spinal cords, so three different isoforms to it. The red are the novel ones that we're also finding in the ALS cord. So now a question becomes, which 3' UTR do we want to test? So we're going to focus on all the rest of the assays. You'll see that we always talk about a short, medium, and a long NFL, and do our assays on that. So the first of this is, what does it mean? And so here's a simple structural thing. Here's the smallest one, looking at the secondary structure. And so this is what we would find in a control spinal cord, right? This is what we find in the ALS spinal cord in addition to this. And it might not seem like an awful lot when you're talking about it, but if you look carefully here at this loop structure, it actually does predict a change in how that stem loop structure is orientated. And for those of us who follow the literature, when we describe the interaction of TDP43 with NfL messenger RNA, we never showed you the binding site. Great paper, but we never showed you that. What we showed you was that we inferred that the binding site was created by stem loop structures coming together to create the interaction site, which was a novel mechanism. The next step would be to crystallize it, and we never did do it. They couldn't do that right at the end of the day. So stem loop structures are really, really important. But if you don't buy that, here's the long isoform, and simply the difference between the two ALS and the control one from control spinal cord, you don't really need much explanation to see that you change that structure dramatically for that. So figuring out which isoform is expressed is also very critical to these assays. So we did that, as I mentioned. We did our luciferase constructs, and we added on each of these. We also did the mouse 3' UTR for reasons you'll see in this very next slide. And so here's the luciferase assay results. And I'm going to spend a minute on this as we work our way through, but they are important. So here's TDP43 done on S, which is the small or the shortest isoform, right? There's M and L sitting in here. And we had previously shown that TDP43 stabilizes NfL messenger RNA. And so we got further example of that here. P190 ROGEF does it as well, stabilizes. This is what Schlepfer had reported. And when we use the short form of human NfL, it also stabilizes, which really fussed the reviewers for our papers because we were showing the exact opposite for RGNEF. And depending on the length of the 3' UTR, greater degrees of destabilization. So remember, this is the human homologue to the murine. It should not have been doing that. So what happened to bring that about? Well, we came across this interesting set of results, which was really that all we were trying to do was figure out the functional domain, and we started at the N-terminus domain and just started whacking off pieces of it, so cleaving into that. And as soon as we got rid of the leucine-rich domain, here's P190 ROGEF as a stabilizer, we completely converted into destabilizing. Didn't touch the RNA binding domain, didn't touch any other, just got rid of the leucine-rich domain, right? And it completely changed its functionality. So it gets worse than that. Right? So P190 ROGEF, we've explained that. Here's RGNEF. And so with RGNEF, and these are a variety of constructs here, right? So here is full length, destabilizes exactly as we expected it to do, right? We got rid of the GEF activation domain, and it had no impact. We were really happy with that. Made sense, it sat here. And then as soon as we got the leucine-rich domain gone, look what happens. Significant increase in its ability to degrade NfL messenger RNA for that. And that is maintained, right, until we get rid of the zinc binding domain. And now the zinc binding domain completely abolishes. As soon as that's gone, the only difference between this construct and this construct, which is here to here, is a zinc binding domain, right? So the important lesson coming out of these studies, right, are these domains, which are sitting distant in the protein to the RNA binding domains, right, are absolutely critical. We need to be thinking about them as we start to think about functionality, right? The zinc binding domain is a critical one in there. So is this important for the protein levels? And this was the holy grail of experiments, for at least in our field, which is that it's one thing to say that you've changed RNA stability. It's another thing entirely to say that that translates into a reduction of the protein itself. And for years in the neurofilament field, we inferred that and then hoped that nobody asked us the question, right? But this is what actually it does do. So here, simply, if we look at a transfection where we put RGNEF in, we reduce the RNA species. That's wonderful. It's exactly what we wanted to see. And here's what happens to NfL protein versus control. Massively suppress it. So it is truly leading to a translational reduction as well. So why is that even remotely interesting in ALS? Well, because now when we go back to ALS spinal cord and do that RNA immunoprecipitation, right, the RNA IP, so we use a protein to bring down the protein species and then do a PCR off that to see what we're bringing down with that. We use, in this case, a GST-fused protein. And what we're doing here is we can bring down, and this is our control for it, we can bring down the ALS NfL messenger RNA. But what's interesting is that it's only, this is a control, but when we start to show you all the ALS, or the cases that are non-ALS, the only thing that we're able to bring down is every time we bring down NfL messenger RNA, it's only out of the ALS spinal cord. We never bring it down out of the control spinal cord. So that's led to this bit of a working hypothesis, which in our lab we call a Tim Hortons hypothesis, because whoever gets it right gets a free cup of coffee at the end of the day for it. And there are two ways of thinking about this. So here's what we're thinking right now with regards to our protein. So we've got our C-terminus and our N-terminus domain here. Here's our lysine or leucine-rich domain here. Here's the coil-coil domain, and here's our RNA binding domain. So one possibility is that this forms a hairpin loop. There's an interaction between some other intermediary protein here, or there's an electrostatic reaction between the leucine and the coil-coil, and the protein is blocked from displacing that, in which case in the ALS phenotype, it's opened and the RNA gets into it. So that's one possibility. The other one is that the leucine-rich domain has nothing to do that, works out here, and for other reasons we have a binding protein here. Same sort of mechanism, except you don't require as extensive a kind of a hairpin loop be made with that. So we're testing this right now to sort it out. But it's conceptually how we're working with this. None of that would be relevant at all if the protein wasn't relevant to ALS. So now we know it's a destabilizer. It leads to reduced levels of NfL messenger RNA, reduced levels of protein. We know that it interacts specifically in the ALS spinal cords with NfL. Who cares? Right? Well, we care when we see it as a pathological protein. It seems to be the way we go with this. So we created an antibody against this, and this is just the proof of all of that. But the next series of confocals I'm going to take you through. So this is a Z stack, and we're going to do deconvolution. So this is RGNEF first in the green. Now I've got P62 in the red, so there's a second antibody with it. The traditional overlay is going to occur right now, so anywhere where it's yellow, you can see the overlay. But now what we're going to do is we're going to subtract away, okay, everything except the top 2% of interactions, right? And so I'm going to get rid of everything in a moment, and this is what's left behind, right? So it's a way of demonstrating that, in fact, you do have co-localization between your two proteins. And the reason for this is that if you don't do a lot of confocal microscopy, you can make an awful lot of mistakes in this. If I have a red fluorochrome in the cell at this level, right, and I'm seeing an awful lot of protein, and I've got a green fluorochrome at this level in the cell, right, remember that's only 10 microns, and you're looking at it, and you're looking at it like this, they will look like they're overlapping, whereas in fact they might not be. So by reducing all the rest of that and getting down to the top 2%, you actually can see those that are truly co-localized, but you'll also see in the subsequent ones, we do co-immunoprecipitations to prove the interaction that's in place. Everybody comfortable with that?
Very much, but I just wonder, what am I looking at here?
This is what would be called, in my day of training, this would have been called skeins, right? So these are anterior horn cells from humans, yeah, with that. Now we just call them neuronal cytoplasmic inclusions for that. Okay, good with that? Because now we're going to get more complex. All right, so this is now looking at RGNEF, first antibody. Here's an RGNEF inclusion sitting within the cell. We can see this extracellular. Here's TDP43 within that cell, large inclusion, right? And now same motor neuron. Now we do the overlay between the two. You can see the co-localization. Next you'll see the white, which is the top 2% of co-localization. And next we're going to subtract away all of the background, showing the absolute co-localization between two RNA binding proteins being expressed in the same cell. Okay, TDP43 and RGNEF. And here's a co-immunoprecipitation just to show you that in fact we can precipitate them down together. So pretty good evidence, same cell, two different proteins, same time. Okay, good with that? Now, how about FUS? So here's RGNEF, all right? Again, we're seeing a cytoplasmic, right? Here's FUS, right? I think we'd all be quite happy that doesn't belong where it is right now for it. Let's do our top 2% co-localization. Now we're going to subtract away, and you're going to be left with the absolute co-localization between the two. Now, those at the front can see these little granules here. That's the axonal process coming through. Now to really make your day, or not, I don't have the Z stack because it corrupted, but here's FUS with TDP43, same motor neuron. Okay, so, and the TDP43 is in red, FUS is in the green, co-localization between the two. And you can see in this particular picture, statically rendered, how these fibrils, these pathological fibrils, are interwoven with each other. Sometimes they're pure, sometimes they're overlapping and integrated with each other. We can bring them down by co-IPs. Right? It's an important concept because we have tended to call TDP40 apathies, FUSopathies, RGNopathies. I don't think they actually exist. right? I think we have a fundamental disorder of RNA metabolism, and these RNA binding proteins can be mal-expressed in the same cell simultaneously and coaggregate. And why do they coaggregate? Well, we heard about that the other day. Here in our world, we talk about them as being intrinsically disordered, but in the yeast world, you can talk about as being prion-like domains. And so here, if we look at FUS TLS, sorry, it's above the screen, but that's all right. Anywhere where you've got a score above a threshold of 0.5 here, these are areas that are considered to be intrinsically disordered, right? Here it is for TDP43, a bit of a shrunken scale because it's such a massive protein, but you can see in RGNEF at least 5 domains that are what we would consider to be intrinsically disordered. And so if I were to show you these one at a time, you'd feel very comfortable saying, yes, and therefore they can form homopolymers. Why can't they form heteropolymers? There's no reason for them to not do that. And that's what we've just shown you in these. So they can be mal-expressed, same cells, they're intrinsically disordered, and they can form these massive aggregates in which they're all incorporated simultaneously for that. Nobody likes to present anything anymore without showing that it's at least inherited. So here's a paper we published last year with a case in which we actually found a deletion or an insertion, both, which will give rise to a massively truncated protein. What's interesting is that that encodes for the leucine-rich domain as well. So what's happening there? But at least we've got a family, and this has now been confirmed in the Chinese, where they have a similar, in fact, exactly the same mutation in that. So how does this then link across to degradation? And I think all of us here doing microRNA work would agree that simply attaching an RNA binding protein on, even though it leads to degradation, it's not the RNA binding protein itself that's leading to the degradation of that transcript. Right? So what else is being brought in? We know that must be the microRNA sitting in that. So this other pathway about which we've heard a fair bit. And it's the work that we did a few years ago in the ALS tissue was while we were starting to ponder this question to ask the next question is that what is the RNA granule composition like in ALS motor neurons? And so these are human spinal cord motor neurons with that. And this one is stained, and they're all actually stained for TDP43, which we see here in what I actually think is the more common picture in an ALS motor neuron, which is this loss from nucleus and this massive cytosolic overexpression. And when we look to see, are they Stauffen, immunoreactive, which would at least link it to transport? Yeah, we got a couple out here sitting in the axon hillock, happy with that. TIA1, which is a stress granule, well, we've got them sitting out here, a few stress granules. This is control. Here, we even have a pathological fibril in here. But when we looked at XRN1, which are the processing body markers, we found this massive colocalization between them. And so then we returned to our RNA IPs, and we brought down each of those granules. And I say that with massive quotations around it, right? But we brought down at least XRN1 immunoreactive and TIA1 immunoreactive or Stauffen immunoreactive, all correlated it back to controls, and then looked at the amount of NEFL message RNA. And it didn't matter whether it was sporadic or ALS, we found a differential partitioning of NEFL into the processing bodies, suggesting that they were being targeted for degradation, which then raised the question about what are the microRNAs that are linked to that. At the time when we first started this, the only thing that was available were the TACMan assays, right? So 645, of which about 30 are control, and then there's LET7. So at the end of the day, you're down to about 600 microRNAs that were actually on those 2 chips at that time. And so we took ventral spinal cord, and so how we did this was we microdissected off the ventral part of the lumbar spinal cord. Since most of our patients, in fact, all die from respiratory, you bias towards more motor neurons if you look in the lumbar region for that. And I simply cut that ventrally, split it in half, and we used it for 2 different assays for it. No more than that in doing this analysis. And then we did the TACMan. And what Denai did in this particular paper was she used a series of predictive algorithms, and she found there were about 103 that would be specific microRNAs for NEFL, but only 19 of them were highly conserved. She then went through each one of those and did quantitative real-time PCR. We did the Luciferase assay to confirm the functionality, and then to a direct interaction by EMSAs. So that was one set of data for that. The next set of data just about killed a postdoc. And this was to develop the human spinal cord microRNA library because it wasn't existent prior to that time. So what we did was in a different set of cases, right, took again the ventral spinal cord, isolated all of the small RNAs from the Mirvana kit. We sent it up to some colleagues for $20,000 later, and a lot of time saved, and they gave us the sequencing library. So that was 3.5 terabytes of data. We then sent that to a group in Belgium who did the microRNA identification for that. We then looked at the novel sequences, and for every one that we thought was related to NfL, we resequenced it by hand and proved by hand using computational devices that they were in fact microRNAs. And then we repeated the same series of experiments. And I'm not going to cause you a lot of pain for that, but this is now on the web. If you want to go and look at the microRNAs of the human spinal cord, whether it be control or ALS based, these are the small RNAs. The microRNAs are these groups up in here. And then we looked at those specifically for our experiments. There's a lot more there, and you're welcome to go for it. This is the bottom line, all right, of all of that. And this is also something that got us into a lot of trouble with reviewers because we identified 5 microRNAs that were differentially expressed between human control and the ALS cases for that. But if we look at the luciferase reporter assays, right, so 146A downregulated, right, so it would lead to a reduction in the amount of NfL messenger RNA when it was put onto luciferase, right? So it's destabilizing it and you're leading to reduced protein. And its expression was upregulated. So that's wonderful, right, with that. But these next 4 all led to an upregulation of expression and protein, which is not what you traditionally think of in human microRNAs, right? But in fact, that's exactly what was happening. And their expression was downregulated in ALS, right? So it's a combinatorial effect that we think we're seeing. And I return back to this slide, which was the slide that suggested that we also have to think about the different isoforms. And as we begin to do this work, so this is really preliminary data, right? As we start to test these individual isoforms, either alone or in combinations for it, we're starting to see that there are significant differences depending on which isoform of NfL is expressed in relationship to the microRNAs as well. A lot of work to be done on this, but it's a clue that that argument that you can't just look at one is going to be sufficient. We need to identify which ones are truly being expressed in there. So that's a lot of data to go through to try and tie together a long story in there, but it can break down into 3 relatively simple, I think, conclusions. One is that a lot of the genes that are associated with familial ALS actually are linked to neurofilament messenger RNA metabolism. I haven't reviewed all of that, but RGNEF is one of them and sits within that. And it performs pathological fibrils, and it's genetically altered, at least in 2 families that we know of. So it's a rare one, but you're going to learn a lot, as we learned earlier from that. This RNA binding proteins, this is an important piece in here. You know, this concept that we call them individual opathies, I think is wrong. I think we should be looking at them as a disorder of the RNA metabolism on whole, and one piece of that is a malrepresentation of the RNA binding proteins, and they can form heteropolymers within a cell. It depends on how hard you look with that. And then microRNAs, I don't think it's going to be, in contrast to some of the other things that we've heard of, where it may well be for tumor biology and the FTDs and such, I don't think it's going to be one. It's going to be a combinatorial effect of the microRNAs at a stage of the disease that's going to determine the stability of NfL messenger RNA. So the complexity gets wrapped up with a lot with this. So these are the people who do all the work. Christian and Denai have been doing, Christian's really led the way on the RGNEF, and Denai and Mohammed. Mohammed's the one we almost killed off with the figuring out the human spinal cord. And they're just all finishing up right now, and are on the market as well for it. And then this is my favorite slide of all, and I won't tell you what it is. You can tell me what you think it is later.